Okay. Hello everyone. So here in this video, I'll be talking about the DNA transformation and the efficiency. So uh, to understand that, I have taken one question which has appeared in 2017 GATE question paper, life sciences question paper from microbiology. So it was question number 72. It is 2 marks question. It is a numerical question. So the question is the transformation efficiency of competent cells prepared in laboratory is 10 to the power 4 CFU per microgram of plasmid DNA. So if 0.01 microgram of this plasma is used to transform these competent cells, the number of transformed bacteria in CFU after plating will be. So before discussing this question, let's first discuss what is transformation. Okay. So what is transformation? Transformation is nothing but allowing cell to take up the foreign DNA. Okay, so suppose you want to work on a gene, you can insert the gene or put the gene into a vector, and now that gene along with the vector, you want it to uh, introduce inside a bacteria. This will be a foreign DNA which you will be introducing in bacteria, and using bacterial cell machinery to produce the product of the gene. Okay, so for these kind of purposes you will be using the transformation. Okay, so transformation is nothing but incorporating the foreign DNA or an exogenous DNA into a cell. So bacterial transformation is where we use the bacteria and transform the cells to it. So that bacteria also naturally, few bacteria have the ability to take up the foreign DNA. So those are the competent bacteria. Otherwise, in laboratory, we have to make the bacterial strains competent. Okay. So the very generalized method to make the cells competent, bacterial cells competent, is using a calcium chloride method, where the calcium divalent cations will be coating the bacterial membrane and allowing the negatively charged DNA to easily come up in the surrounding of the bacterial wall, and when provided a shock, the bacterial cell can easily take up that DNA, and then we allow that cell to recover by adding some media and then plate it on a antibiotic selection plate. So with the antibiotic selection, the bacterial cells which have taken up that foreign DNA, they will only grow. So because antibiotic will gonna kill the bacterias which are non-transformant. Okay, so but when you transform, so along with the plasmid, you are also sending one gene which is providing resistance against the antibiotic. Okay, so if the bacteria has taken up that DNA, so that bacterial cells which has taken up the DNA, they will resist the antibiotic pressure. Okay, so they will grow, they will survive on the antibiotic plate. So that is the transformant bacteria, or you can say CFU, which is nothing but one bacteria makes the colony, and that refers to as colony forming unit. Okay, so the efficiency is measured as the number of transformant per microgram of DNA added to the cell. Okay, so transformation efficiency CFU upon concentration of DNA plated. So you need to know how much you have plated. Maybe you have added initially some amount to transform, but what you are actually plating, that you need to know. So to know the concentration of DNA plated, you need to know the amount of DNA you use for the transformation, how much volume you have actually plated, and what will be the recovery volume you have given. As I have mentioned earlier, that you add some amount of media so that recovery of the cells can happen after the heat shock and cold shock. Okay, so the ultimate formula comes that number of transformants into final volume at the time of recovery, microgram of DNA, volume plated, and number of transformants. Okay, so suppose you got 100 colonies and you have final volume was 1000 not 1000, it will be written in ml. So 1 ml. While plating, you have given 0.1 microgram of DNA and you have plated only 100 microliter, that means 0.1 ml. So if we'll calculate this, it will be, it will come as, so it will come as 10,000, okay, 10,000 CFU per microgram. That means if you are transforming 1 microgram of DNA, you will be getting 10,000 colonies. Okay, that is the simple meaning. Okay, and if you are transforming, okay, 0.1 gram and plating 100 microliter out of it, like this. So if you are transforming 0.1 gram and plating the entire thing, then you will get 1,000 colonies. But here in this case, even though 0.1 gram has been transformed, but only 100 microliter out of it is plated. That is why 100 colonies has been got. Okay, so this is the calculation. So if we'll see our question, and our question also, it has been said that the transformation efficiency of competent cell prepared in the laboratory is 10 to the power 4 CFU per microgram. Okay, that means when you are giving 1 microgram of DNA, you are getting 10,000 transformants. So similarly, it has been asked if 0.01 microgram of this plasmid is used, then how many transformed bacteria or CFU will be observed. So it will be a simple calculation, multiply, and then you'll get 100 CFU. So if you are transforming 0.01 microgram, you will get 100 CFU. If you are transforming 0.1, you'll get 1,000. If you are transforming 1, then you'll get 10,000. So that is the simple calculation. Okay, so this is it, very easy to solve. And this is my NPTEL reference where you can understand the DNA transformation protocol, where in the laboratory you can see how it has been performed, and it will be easy to understand. Thank you.